Today, Dr. Jyoti Sharma. Dr. Sharma did her bachelor's and master's in biotechnology in India and worked at the Punjab Agricultural University with rust diseases. She then completed her PhD degree in plant genomics and bioinformatics from North Dakota State University with Dr. Justin Ferris and Dr. Stephen Zhu, and focused on the genomic analysis of wheat domestication and rust diseases. Dr. Sharma worked in agriculture and agri-food Canada as a postdoc and term researcher on wheat rust resistance and suppressor genes. She joined University of Minnesota in 2022 as a postdoc with Dr. Pablo Rivera and Dr. Eugene. She's currently working on wheat and Barberry genomics, and now the floor is yours.
Thank you. Okay. So thank you for introduction, and I'm Jyoti Saini, and today I'm going to present our work on development of molecular diagnostic tools which can help to detect the hybrid Barberry. And we are also presenting our results to investigate the presence of the hybrid Barberry and other Barberries around the Minnesota state and testing them for the stem rust reaction. So before going ahead, so what the mean of the hybrid Barberry? So hybrid Barberry here we are presenting is the plant which is produced by the combination between the common Barberry, which most of the people working in the stem rust known as the alternate host, and the Japanese Barberry, which we can observe in different landscaping and another issues which we have in the invasive species. So the interspecific hybrid Barberry, which is developed by the cross between these two plants. So that's the one of interest of today. With that, so first a little bit background about the common Barberry, why we are concerned about that. So common Barberry, which is also known as Berberis vulgaris, and it has been introduced by the early European settlers in the North America region, and it's known to be invasive. And if we know about the wheat stem rust history in the here, so we know that it has been eradicated in the most of the part, and this program has been run for multiple years. So in most of the part, we can see in the Minnesota, we observe that Minnesota has the Barberry has already been eradicated, but on the basis of the ED maps, there are a few locations in the North America where we can still see the common Barberry present in different states. And now we can move to the next plant. Before going to the next, as I mentioned, how we are concerned about the Barberry and how it's related with the stem rust, as we can see here. So the Barberry plant is work as the alternate host to the stem rust pathogen, so which can help in the sexual cycle and which will lead to the production of the new races. So that's the reason it has been eradicated during the epidemic of the stem rust in 90s, 50s or 60s. So this is the one of the main reason about the common Barberry eradication. And now we move to the next, the Japanese Barberry. The Japanese Barberry is another plant that has been introduced in 1875, and it is a replacement of the common Barberry for the landscaping and for the other, I would say the horticulture industries. So this is the one of the Japanese Barberry we see in the campus in near the housing complex, and you can also see in the front of the student union. We have lots of the Japanese Barberry plants there. So they has been used in the landscaping, so they are common around. So another thing here we are presenting. So Japanese Barberry is present in the different location across the North America, especially if we can look in the East Coast. There's multiple plants has been reported for this Japanese Barberry. And there are some reports you can see where there are some ban on this Japanese Barberry, which is also known as invasive, and it has been a role in other different diseases, tick disease, deer tick disease. So that's the role of these two plants. So now we know the background, and now we move towards the hybrid Barberry. So, and we say that the hybrid barberry, which is present, they can naturally easily cross these two plants if they are present in the environment. For example, in the East Coast, these two both parents are present, so they can easily hybridize and they can produce them. And there also has been created in the control environment. As you can see here, there is some East Coast and in Minnesota region, the hybrid barberry detection has been reported on the EdMaps. So now, why it's important to detect these hybrid? Why we are concerned? The first main reason, they are invasive. Their fruits can easily spread. As we can see in this figure that has been collected in the Osprey wilds, where you can see the multiple plants are present within the very short space. So they can replace the natural environment and they become invasive. And then the soil morphology, they can easily change the soil morphology. So that's the one main reason to look for these plants. And if we look from the stem rest and the food security point of view, the hybrid has the ability, so they can host the stem rest pathogen. So if they can host it, they can let the pathogen complete this sexual cycle, so there is new races can come forward. So that's the reason we need to look for these plants. So the next question, how we can detect this hybrid? So whenever we are saying we have the hybrid plants, so did they look phenotypically different from the parental species? Or what any genetic tools we have available, so which we can help to detect them. So let's go with the phenotypic thing first. So if we look here, we have the common Barberry. So there is some features we can easily see in the figure. So the first thing you can see that I think in this figure is not pretty clear. So the Barberry has the serrated leaves. But in this figure, you can see the Japanese Barberry has non-serrated leaves. So very obvious, like phenotypic feature we can see. And they have the different inflorescence where you have lawn, Panatin, erasemes, and they have the solitary flowers. So that's another common feature in this one. So the common Barberry has multiple spines, but for the Japanese Barberry, we have one spine per node. That's not visible in this slide. So now we think about, okay, what the hybrid will look like. But when we look at the hybrid, there is huge variation in the leaf morphology. We will discuss more how much variation in the coming slides. So there is a variation in the spines per node, and they have different types of fluorescent structure, but this thing you cannot always observe when you're going outside in the woods, you're looking for the plants. So in short, we can say that the morphological traits are not always diagnostic. We need to come up with some molecular tools so we can detect this plant. So with that, so this is our main objective to conduct this project. So first thing, how we plan to achieve our target. So the first thing we need to survey across the Minnesota to look, is this plant is present across the Minnesota? And we also did the rust evaluation, the plants we have collected during our survey. We developed the diagnostic markers and we test them. And we test the plants which we have collected and live tissue samples. We will discuss more in detail in coming slides. And another thing we have did, the F1 hybrid population cloning, and we also did the rust evaluation. The importance of F1 population, we will explain later when we are saying there is huge morphological variation. We will be able to see this in this population. So when we started to do our surveys, two things we keep in mind. The first, we try to look all the location around where we can go in the state parks and the different forest areas. But we also keep in track on EdMaps. So if there is any report, we definitely cover that location and look for the different plants in that region. So that's how we plan our surveys. So in 21 and 22, I was a part of the project in 22 survey. So we look across the different counties and we collect the live tissue samples. So in 2021, we are able to collect 171, and in 2022, we collect around 132. So the Vulgaris species here is, we have the one known location, the Bath farm, and we have some other sample collected across the Minnesota. So in total, we have also collected some live plants across our survey trips. So this is a outline of our survey trips. So during our survey trips, we are not able to see any rust, but we are able to see the necrotic spots, and we are working on to identify more on these spots in the coming month. And we will again doing survey in next month or by June to see this location again to see if we are able to see any rust around. Now, as I mentioned, we collect the live plant. So here we will give a little bit background of what kind of variation in the morphology we have observed. So in this picture, we have two reference images. There, one is the common Barberry. You can see clearly the seriation on the leaf structure, which was not present in the previous slide. And in the Japanese Barberry, we have very smooth leaf surfaces here. So now when we go for the survey trips, we have seen huge variation in the morphology, leaf morphology in 2021 and 2022 survey samples. As you can see here, we have leaves that range from very small to very large leaves. We have very different shapes, heart shapes, some are wavy. That's clearly not looking like a Japanese Barberry if we can compare them. And we have the spines on the leaf surface. So this is the one leaf you can see here from the Afton State Park, which has serrated surface. And these ones from the Osprey Wild, that's in Sandstone, Minnesota region. So these are the location you can see, we see the huge variation. Now, when we say we select the live plant, how we choose that? So first, when we look the plant, we look for the leaf morphology. And we also look at, you can see here, you can clearly see the 3 spines, but a little bit more clarification. So we also observe the multiple spines per node. Here you have 5 spines, 4 spines, which is not common in Japanese. In Japanese, you have always 1. So that's classified them into the putative hybrids that we need to take them back. And we I think only 2 times we observe in 22 the flowers, but we take the notes, whatever we are noticing down. And then we dig it out. We bring it back to the lab. And then what's the next step? We did the rust evaluation on these plants. So we already finished our 21 plant, 21 survey samples, but we did evaluation in 22. So these are the plants. So here is the positive check, chinensis. Before going to the results, this is our standardized method in the CDL. So where on the top, we have the stem rust infected wheat straw. And then we have the plants, and we maintain the humidity for 4 to 5 days after the infection. We put the plants outside, and then we observe for the reaction. So this is a method we used, and we have observed in 21 samples, we have necrotic spots. I'm not presenting here, but in 22, we also observed the necrotic spots on few samples. So within few days, we are completing our next set of rust evaluations. So now we notice there is rust evaluation, but there is no sporulation we have observed till now for the live samples. Now this is, I mentioned in 22 after survey, we have observed the necrotic spots. So this is the one picture I presented before. And we can see the leaf morphology here, which is completely different. And then we have also, we can see the necrotic spots we have. These are currently ongoing. So they have rust inoculation. We are seeing the necrotic spots on the different leaves. So all these things suggesting us there is maybe the presence of hybrids in the Minnesota. So now we know we look for the phenotypic traits, how variable they are. Now the things comes up, the diagnostic molecular tools. For that, we use the previous study data, which is conducted by Radhika, and she was working on this project before me. And they have the GBS study, which was conducted, in which they have used the Barberry, common Barberry, Japanese Barberry, and some hybrid. So based on that, the final outcome, we are able to find the 33 paste markers, Smith-based markers, so that can detect the hybrid allele in the Arboretum collection. So this Arboretum collection has multiple Sebaceae and Berberis collections, and they also have some known hybrids in the region. So that acts as our validation panel that can tell this markers can detect. And this is the one image where you can see we have the vulgaris allele, and then we have the Japanese Barberry, and we have clear hybrid here, where we also use the F1 as a control. So the clear cluster formation, we use this marker to move forward on our survey samples. So we took these 33 paste markers, and this is a little bit more into detail of how they work in the Arboretum collection. So in the first set here, we have the vulgaris and the both parents, Subertii and F1. So that the set of the population from where these markers were developed, you can clearly see the actually here. And now we come down to the vulgaris we have in the Minnesota. So these are we collected from the Bath Farm and the Detroit Lake. And then we have the Japanese Barberry from the different location. This is the one we collected from the Student Union. They also have the Japanese Barberry. So this show the validity of this marker, so they can work across different barberries collected in different states. And also there are some known hybrids, so where we are able to detect, these markers have ability to detect the hybrid alleles in these ones. In addition to that, it also have ability that we can see the chinensis and vulgaris have the same A allele. So that's the next step we need to work on. But so that's the results we got from our initial analysis. With that, this is too much overwhelming picture, but I will try to cut short it. So in 2022 and 2021, these are the different locations we visited in both years. And these are the 33 paste markers that work on my Arboretum collection. So if there is any colored column here, that means this marker has detected hybrid allele from the samples from this location. So in this one, we can easily see that some markers can detect the hybrid in different locations. For example, 61, it was able to detect the hybrid allele in different locations for both 22 and 21, and 98, 122. So that shows the utility of these markers if we are going out for survey trips. Now, when we coming down out of these 33, we come down to 10 paste markers. But that doesn't mean like we are excluding the others, but maybe in the next year when we are going out, we again will test these markers on the same set of markers to see if we are able to detect more. So here is the more visualization of how this is our 22 survey samples. We have different markers and how we are able to see the H alleles in different samples, and you can easily see the clear cluster formation, and we have the positive and negative controls with the sample cluster formation. So now we have the phenotypic results and now we have the genotypic results, but if we are looking how they are coordinating with each other. In this one, I'm presenting two examples here. So this is our 22 survey sample, 70 that has been collected in osprey wiles. You can see the necrotic spots, and we also, our marker is detecting the hybrid allele in this one. And then we have the plant that's collected from the afton state, and it doesn't have the H allele. So when we coming to this, then we cannot only rely on one feature. We need to look at the overall picture because this is the first step when we're going towards the genotypic stage to analysis these hybrids. More things will come up, but we need to look for the overall picture here. So what we know in the altogether from this survey. So there is definitely phenotypic variation in the all the barberries around in the Minnesota region. And we are able to detect the hybrid allele. This is one culture. We have the multiple spines, leaf structure is different, large leaves, and markers are also telling there is hybrid. So with that, what we are delivering with is our outcome of this project. So we have the Barberry DNA extraction it's complicated. They have lots of factors comes up with this one. So our procedure is standardized procedure, which can be used on the different Barberry species to extract DNA. We have narrowed down to 33 paste markers, and this protocol can be adapted to different labs to work on this one. With that, now we move towards the F1 hybrid population. why we need to go back on this one, and what's the significance of this population. So this population is developed in New Hampshire, and we are planning to, we bring this population here, and let's go to that one. So why we need to look this one? First, it will help to understand what kind of morphological variation we're gonna see in F1 population, and what kind of phenotypic variation, especially the stemless reaction you can see in the F1 hybrid. So to bring this population, we bring the cuttings from New Hampshire. We are able to retrieve around 65% of this population. And then we did the stemless screening. In this one, we have the parent common Barberry. You can clearly see how infected it is. And we have very clear Japanese Barberry with zero reaction. And then we have the F1 hybrid population leaves in middle. One thing we can see here, leaf morphological variation, how they are varied from within, even if it's F1 population, they are varied for the stemless reaction. Let's go more into the stemless reaction. So in this one, if you can clearly see here, the common Barberry and the Japanese Barberry, there's a high form, Pyxnia form, and there we have the acial cups has been formed. And we have the leaves here where we have only necrotic spots with few Pyxnia and only necrotic spots. So that's explain how much variation we can see in the F1 population. So if we are going in the environment, you don't know what stage the plant you're looking at. So you don't know what expectation you have. You cannot rely only on the leaf structure. You cannot rely only on the stemless reaction. You need to look overall picture on this plant. So now another thing came into mind. If we are looking at the natural environment, we don't have only the common and the Japanese Barberry. In environment, we have different Barberry species and they are compatible to make process. So we need to think about, we are taking the plant and we are testing the markers which should develop in one. So we need to go beyond that. So with that, going back into the thing, why is like we are saying Barberry and this hybrid when I'm saying the stemless thing again. So in the multiple reports in the Europe, we are saying the Barberry playing important role. So that is important. Their new races are coming, has been identified. So that's explained. They have, we need to look into this one again. And with that, I would like to introduce our future direction in our lab, where we are bringing this project to the national level and with the agreement with USDA and University of Minnesota to test these hybrid Barberry on the national level to see how we can utilize these methods. And with that, I think it's very important to understand this hybrid and Barberry and everything if we are thinking about overall picture, where we are dealing with all host, alternative host, and pathogen. And our lab work on all three, but today I present only Barberry perspective in front of you all. With that, I would like to thank our funding resources to provide us fund to conduct this research. And our collaborator, Iago, helped me to teach me about the cuttings and help us to bring plants here. And the initial this GBS work has been done in New Hampshire. And with that, I would like to thank my lab members. They are here. Thank you so much.
Actually, I was trying to understand what are these case markers and how they have been developed.
They are like cast markers. So they are like same, but it's a different company. They're a little bit inexpensive in that sense, but technology is fluorescence based. They are field-based markers. Yeah. So they have different their own like paste mix. So that's all. They are SNP based. Yeah.
Has Barberry been sequenced any of the species?
Only one or two has been sequenced. That has been sequenced in New Hampshire. They use that for initial studies, but that will following up on more of that because there is no standardized. Nobody has a report like of the pure Barberry. I think that's what Eugene can explain more the problem on that.
studied for a while.
So these 23 markers, when it was developed, was only one species of Barberry was used or hybrid were used?
So the first, when they did in the New Hampshire, so what they have a multiple common Barberry, like 12 common Barberry, they have some Japanese and they have some hybrid. From there, they come down to some markers. And then those marker, out of those, we select around 120, the co-dominant. So there are multiple species, not only like two common, like only one common Barberry and one Japanese. So there was like 12, and I think another one was around 10. So both species were that range in that one. Yeah.
So because here, not like you have to use all the 23 markers to really say if it is hybrid or not.
Yeah, so there is another thing. So we have that 120. So out of 120, I was come down to 95 that can actually, like if we have F1, so that 95 work. For 95, I cut down to 33. So that was able to detect all the Barberry and other hybrid from our arboretum collection. So we are thinking about national level where we are having Barberry from across different different states, and we have Japanese Barberry to see how these markers will work on those ones. So we are cutting down to 33. And let's see if we are conducting more study and GBS will be coming down with more numbers.
So what is, sorry,
No.
So what's your cutoff to say that, okay, let's say I have 23 primers, and then if these many are positive, I will call it hybrid, or I will call it the parental.
If I would say, even if the out of these 33, if I would say 1 or 2 will be enough, but if we are saying, because we need to think about that, because we are only dealing with very few loci in this region, but if we are, there's a different approaches I think we need to take. That's what my understanding to take forward this. So I would just consider it's like a first step to better understanding the hybrid. Lots of I think we're discussing in lab now, we are coming up with more topics, but I would test all 33. And before saying like what is hybrid and not, I will look into other features too, especially if we are collecting samples, like for example, in Afghan state, there was a location, you have hundreds of these plants. So you need to look overall picture of that location. If maybe you have one plant which has no hybrid allele, maybe the other plant has the hybrid allele. So I was thinking there's more thing need to be done. As you say, like Eugene said, like 0 thing has been done, we will come up with more.
Thank you.
Thank you very much for the nice presentation. My good colleague here, Eugene, shared with me a troubling document one time that said that they weren't going to evaluate new barberries going into the horticultural trade. I wonder if you could give me an update on what the status is on that.
I think that's a regular we did every year.
No, this is something else.
Oh, okay.
The USDA aphids regulatory Barberry as law. After the that we test the stemless resistant before going to sell in the commercial industry. That's the from Barberry eradication inventory and the act. This law you have to do this. So we do Ross testing here, provide this certification. Now with various pressures, it's expensive to do it. Without stimulus production for decades, the question was relevant to doing this. That's where the question raised we taking this law in book and some of the people.
Do you have a current status of if they're going to continue to fund it, or are they going to drop it?
Well, this year was the first year that national improvement brought that into lobby. You can see, yeah, this was very high up in this
I don't know
Nothing official.
that. Yeah. Because I know we tested and we did it work, but background, I don't know.
So sometimes these hybrid species, you see different reproductive barriers between the different parent species. Do you know anything about the fitness of it? Does it have seed viability, things like that?
I heard it's mostly like they are very compatible easily, like in the environment, but there are some varieties. That's what like government has put restriction that you need to like the variety that are not like producing a viable seeds. That's the one. I'm not sure a different variety that has been in horticulture, but not the one we are talking here. But there are some Barberry species that if they're coming into the sale, they have to be that feature need to be included in the plant. Yeah, but they are very compatible species. Not even like a common and this Berberis, even the chinensis, they all can easily like make cross with each other. So that's why we are thinking when we are collecting samples in the natural environment, we don't know either they hybrid between the Berberis or Berberis, or there is like other Barberry species around there. So that's the things in the mind we need to consider too.
No questions on Zoom. Great presentation. Thank you so much.
Thank you.